JAK-STAT signaling pathway by Elkovich. The JAK family consists of 4 members, JAKs 1 through 3 and tyke 2. JAK stands for Janus kinase and tyke for tyrosine kinase. You'll recall from cell bio that kinase participate in the transfer of phosphate groups. JAKs are protein tyrosine kinases, or PTK, that each associate with a different set of membrane receptors. Until activated, JAK proteins remain dormant within the cell. Once activated, these kinase proteins phosphorylate themselves and the tyrosine residues of their receptors. The STAT family is somewhat larger, consisting of 7 members, STATs 1 through 4, STAT 5A, STAT 5B, and STAT 6. STAT stands for Signal Transducers and Activators of Transcription. By this definition, these proteins are obviously involved in signaling and transcription. STATs dock themselves on the phosphorylated tyrosine residues of the membrane receptors. From this position, JAKs will phosphorylate STATs and send them to the nucleus to initiate transcription. Now let's see it in action. Here you can see a variety of membrane receptors, each with their own associated JAK proteins. As the receptor-specific ligands bind, a conformational change occurs. This spatial orientation activates the JAKs, which cross-phosphorylate each other. They also add phosphate groups to the tyrosine residues of the receptor, producing a docking station for the STATs. When the STAT proteins connect at the docking station, the JAKs transfer the last phosphate groups to the STATs. The phosphorylated STATs dimerize and then travel into the nucleus. The STATs act as a transcription factor as they connect to unique regions of the DNA and initiate gene transcription. Depending on which JAKs phosphorylate which STATs, different proteins are synthesized within the nucleus. So, as a review, in fibroblast growth factor, or FGF signaling, a variety of surface receptors are inserted in the membrane. Specific FGF ligand molecules bind to their corresponding receptors to initiate a series of events within the cell. The end result is the expression of certain proteins through selectivity of activated transcription factors.